In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum, dear students. How are you all? I welcome you in Pakistan International School Thai's virtual learning system for the session 2020. And this is biology class for grade first year. As you know that we have started chapter number 4, and today we will see last topic of chapter number 4, that is about C4 photosynthesis cycle. Then we will start chapter number 5, that is section number 2 about biodiversity. So we will see in chapter number 5 acellular life, and then today we will see viruses. So let's get started. Dear students, C4 photosynthesis or C4 cycle of photosynthesis, that is actually an adaptation to the photorespiration. This problem exists in plants in very hot temperature and as well as in tropical climate, as we have seen before in the previous lecture about C2 cycle, in which plants undergo into photorespiration. So this is actually plants adopted against C2 cycle. So plants adopted a C4 cycle. So the plants who got this C4 cycle of photosynthesis, that is called C4 plant, and the process is called C4 photosynthesis process. The plant whose first product of photosynthesis is carbon compound, so that is called C4 plant. Like in the cycle, like in C3 cycle, in Calvin cycle, the first product that was a stable product, that was a C3. So that's why it is called C3 cycle and C3 plants. So today we will see C4 plants, in which the first stable compound is C4, which is having 4 carbons. So why plants use C4 mechanism? Because some plants which live in drought condition at high temperature and nitrogen and carbon dioxide limitations, so they use C4 mechanism for their survival. So they use like C4 plants fix more carbon dioxide as compared to C3 plants, and C4 plants lose 277 molecules of water to fix carbon dioxide molecule, as compared to C3 plants, which utilize 833 water molecules. So that is comparatively better adaptation for the plants, because they live in tropic environment and very hot temperature, in which the plants need to save their water content, so they avoid photorespiration process. So plants or C4 enzyme fix carbon more efficiently as compared to C3 plant, who fix uh, also oxygen during carbon fixation process. So fixation of oxygen start the process of respiration. We know that instead of carboxylation. So in that substrate oxidized, and the result loss of plant, loss of process, loss of respiration. So this process occurred due to the dual activity of Robisco in C3 plants, such as Robisco is ribulose bisphosphate oxygenase and carboxylase activity. So that all we have studied about photorespiration before. Here is one very important point regarding this C4 photosynthesis is kranz anatomy. Kranz anatomy actually, that is derived by Kranz, the scientist who just discovered or described that anatomy of the arrangement of the mesophyll cells and cells in the leaves of the plant. So here you can see in the diagram, this is lower epidermis, upper epidermis, in which the stomata are present, and these are mesophyll cells. Now mesophyll cells are arranged in the circular structure, and in this circular structure bundle sheet cells are present. So their vascular bundles are surrounded. These are vascular bundles. They are surrounded by rings of cells. So the inner ring is called as bundle sheath cells, and they are starch-rich chloroplast, which like a grana. If there are grana, so there is photosynthesis, but here there are no granas. So what they do, we will see in the next slides. Outer ring contain mesophyll cells, then which go to the photosynthesis. So, what is the advantage of kranz anatomy? That main function of it is to provide the site for in which carbon dioxide concentration around the rubisco is more to avoid photorespiration mechanism. So, how this all is happened, we will see in its mechanism. The first step is conversion of PEP into oxaloacetic acid. So in this step, carbon dioxide react with phosphoenol pyruvic acid to form oxaloacetic acid. So from air, when carbon dioxide enters into the mesophyll cells, so in the leaf, as we know that in kranz anatomy, mesophyll cells are around this one, and inside here we are having the bundle sheath, and here we are having um, vascular bundle. So from outside, when the air enters through stomata into the mesophyll cells, so carbon dioxide react with phosphoenol pyruvic acid, because glycolysis is going on over there, to form oxaloacetic acid. This oxaloacetic acid is actually, what is that? That is C4 molecule, C4. That is C4. That's why this is called as C4 photosynthesis. So this reaction takes place in the presence of enzyme phosphoenol pyruvate carboxylase. That is the enzyme of which perform the fixation, as you know, carboxylase. So it means it performs the fixation of carbon dioxide. So in the second step, we will see conversion of oxaloacetic acid or oxaloacetate into malate. This oxaloacetic acid is converted into malate, and then this reaction is catalyzed by malate dehydrogenase. Dehydrogenase. So what will happen here in this reaction, that is also called a reaction in it means uh, the removing of the hair NADPH2 is oxidized, like hydrogen is removed from that. Number third step, conversion of. So in this step, conversion of malate into pyruvic acid. So now malate is transported into bundle sheath cells. In bundle sheath cell, it is converted into pyruvic acid by releasing carbon dioxide. Now carbon dioxide released in here the inside cell bundle sheath cells from the mesophyll cells, it comes carbon dioxide is released in the bundle sheath cells. Then in fourth step, we will see conversion of pyruvic acid into PEP. The third step was said to be carboxylation. Now in fourth step, pyruvic acid react with ATP, and then it will regenerate phosphoenol pyruvate, and this phosphoenol pyruvate um, takes place in the presence of pyruvate orthophosphate dikinase. So it means phosphoenol pyruvate again regenerated, and this will move again. That is called PEP that will again involved in the carboxylation reaction. So the first one was carboxylation. We will see here in this reaction. Like in atmosphere, carbon dioxide comes into what we say the chloroplast in the mesophils, from where phosphoenol pyruvate carboxylase, that is enzyme which convert phosphoenol pyruvate into oxaloacetate. Oxaloacetate then, in the presence of malate dehydrogenase as well as NADPH, will be oxidized into NADP and it will convert it into malate. Here this reaction is called carboxylation. And then the second step we will have reduction process. The third one, when this malate is converted into what we can say the pyruvate by changing like this is called a decarboxylation here, where a carbon dioxide is removed out by reduction of NADP into NADPH. This is called decarboxylation. Now this carbon dioxide will carbon dioxide amount is increased in this reaction, and now it will move into Calvin cycle, in the Calvin cycle, and then whole the Calvin cycle will move on and the formation of glucose will continue, like photosynthesis continues in the reaction. The pyruvate, which is formed by malate, that will again move into the what we can say here into the chloroplast, and then into the chloroplast in the mesophyll cells, in the mesophyll cells, and pyruvate phosphate dikinase again change into PEP. So pyruvate will regenerate again and again into PEP, phosphoenol pyruvate, and then this reaction will go on, and carbon dioxide amount here it is going to be increased in bundle sheath. From bundle sheath, these if you have seen mesophyll cells, then PEP carboxylase enzyme is involved. Carboxyloacetate, that is 4 carbon compound that is formed, then change into malate, and then in Calvin cycle it carbon dioxide will move into and pyruvate will move again into that mesophyll cell. So here carbon dioxide will go into the Calvin cycle, and then into the sugar which will form, that will go into vascular bundle. So here that is done in the cytoplasm. That's why um, that can be done in bundle sheath also. So dear students, by this we have completed our chapter number 4, and as you can see, the C4 plants are maize, sugarcane, millet, sorghum and grass species are there, which are monocots mostly, and they do this type of photosynthesis. Dear students, today we will also start section number 2, biodiversity. In biodiversity means the diversity in living organisms. So we will talk about here first chapter, that first chapter of biodiversity, or overall that is fifth chapter, acellular life. So in acellular life we will see about viruses, viroid, satellite and prions in detail. What are these? Acellular life, in this viruses are having DNA and RNA encapsulated in protein core, and we can see no ribosomes, and cannot be included in the 16S Sedberg ribosomal RNA. Tree of life, that is includes phage viruses, bacteriophages, and lots of other infectious viruses which acts on bacteria and other organisms also. Viroids are the smallest non-infectious agents, and they are having circular single-stranded RNA in it, and RNA polymerase 2 catalyzes rolling circle synthesis of new viroids. So that is actually mm, that is, we can say, also the present in our body, and they are without protein coat. Satellite, they are subviral agents composed of just nucleic acid, which depends on co-infection of a host cells and with the help of a master virus. So they need some other virus for their propagation. So prions, they are also the proteins which can fold in multiple structurally distinct ways, at least one of which transmissible to other prion proteins. So that is actually infected protein which are present in our body. So they can be converted into prions. So today we will discuss in detail about viruses. Viruses are also called virions. They are having different characteristics. If we see their characteristics, so they are living as well as they are non-living. So how they can be distinguished? That they reproduce at a fantastic rate but only in the living host cells. So their genetic material can be in the form of DNA and RNA, as well as they can mutate in different forms also. So they can cause lots of diseases, and they can be intracellular obligate parasites. They get destroyed by ultraviolet rays also. If we talk about non-living characteristics, so they are acellular, they contain no cytoplasm, no cellular organelles, so they carry out no metabolic activities on their own, must replicate using host cells for their metabolic activities. They do not respire, and they do not grow and divide in the absence of the host. They possess DNA or RNA, but never both. They can have just DNA or just RNA. They do not have a cellular structure, um, so which is the basis of life. So they do not respire. Viruses behave as non-living inert infectious particles outside the host. So if we just see their history, so viruses are actually, we can say, the derived from the Latin word that meaning poison, name virus, and that meaning is poison, or that is also called venom, that because they are infectious in nature. So the first evidence was like Stanley in 1935. He has crystallized a sap from stick tobacco plants. So he just worked also on tobacco mosaic virus, and he gave like in 1935, like he crystallized that virus, and that is was, we can say, the first discovered virus, that is tobacco mosaic virus, TMV. So he discovered that virus which are having nucleic acids and proteins, that is TMV. The study of virus is called virology. So in 1885, Louis Pasteur, he also worked extensively on rabies. So he identified as that rabies is caused by virus from the Latin word poison, but first he didn't give the name of that. Louis Pasteur worked with Cumberland, and they discovered the causative agent of rabies to pass down from porcelain filter. Porcelain filter is like the filter which is having pore sizes almost zero point 0.1, 0.1 to 1 micron. Okay, so that can be 100 to 1000 nm nanometer. So in spite of this, he did not discriminate between bacterial and other agents of disease. So actually he cannot distinguish properly that bacteria cannot pass through filter paper, but the abstract which he got after the filtration, so uh, he just saw that filters could be used to completely remove all bacteria but other cells known at the time. From the liquid suspension he got some other substances from that liquid which he extract got from the filtration. So in 1892, Dimitri Ivanovsky that he described that the filterable infectious agents, that is in tobacco mosaic virus, so they are smaller than any known bacteria. So he was the first person who discriminated between viruses and other infectious agents like bacteria. And later on, we have discussed that Stanley just did this crystallization of this virus, and he got Nobel Prize in 1935. So that was all about viruses, its introduction, characteristics, and discovery of viruses, as well as history. So, dear students, that's it for today. Inshallah, we will continue tomorrow with the classification of virus. Wish you all the best. Allah Hafiz.